What is coevolution? Well, in the simplest formulation, it's really the reciprocal evolutionary effects that one species has on another species. And basically, through this interaction, evolutionary change happens. Species A evolves in response to some selective pressure by species B, and vice versa. Species B, because of this interaction, also could respond to the presence of species A, some activity, some interaction that species A has to alter the fitness of species B. So when does coevolution occur? Well, the definition would say that those two species need to be interacting. And so the closer the ecological relationship, especially with a fitness difference, the more likely it is that coevolution will actually occur. And usually this kind of close relationship happens between species that are specialists rather than between species that are generalists. This is a topic, this is something that we're going to come back to later on. And it's important to differentiate between the measurement and the study of coevolution at two different scales. One is on an ecological time scale, that is within a generation or several generations, and measuring the effects that species have on each other. And one is over evolutionary time, something that may happen over not only hundreds, but maybe thousands of generations, over perhaps even millennia. And so at one end of the scale, you may be measuring things like survivorship, host plant selection behavior, traits that may change and that make insects or plants susceptible or resistant to each other. And on the other end, you may be experiencing or you may be measuring things like the evolution of specialization, why things have broad diet breadths, diversity of lineages, and so on. We're going to take each one of these two in turn today. The roots of the study of coevolution really started about the middle of the last century. Some of the best examples that emerged at the time were having to do with what's often referred to as a gene-for-gene interaction between plants and fungal pathogens. That is, there was a pathogen and a plant had a single trait governed by a single gene that resulted in the resistance to that particular fungal pathogen. Because of the existence of that resistance trait, fungi that had traits themselves that allowed them to overcome that resistance became adapted to that new type. And so there could be this gene-for-gene change between the host and the pathogen that allowed them to evolve in response to each other, breaking that resistance in a sense. So these ideas were floating around in people's minds as tools to study the plants and their chemistry were becoming more and more common. As you heard in a previous lecture, Gottfried Frankl, in trying to understand why there was this diversity of chemistries within plants, came to the realization or proposed an hypothesis that said that actually the reason for the existence of these plant secondary metabolites was not because of some garbage waste products that they were creating, but it was actually because they conferred some level of defense against important selective agents like herbivores. So Gottfried Frankl proposed basically a mechanism by which plants could actually defend themselves against insects and therefore could have a selective advantage over others in a population that didn't have these particular traits. Ehrlich and Raven proposed a modification or kind of a next step from this in a classic study that was entitled Butterflies and Plants, a Study in Coevolution. And in this particular paper, they proposed a series of potential steps that through the interaction between plants and insects, you could get the evolution of different defensive chemistries and also the speciation of lineages, the diversification of lineages to the point where they could no longer interbreed. And therefore, the radiation and the explosion of, and therefore the generation of new species because of the response of a plant to an insect, for example. here's what Ehrlich and Raven actually proposed. They basically said that either by mutation or recombination, just by some chance event, or by the mixing up of traits or of genes that were already there, angiosperms, that is flowering plants, could produce some new plant substance, secondary compound. Again, these are compounds that are not serving a primary physiological role, but can serve these additional roles, such as by mediating interactions with other organisms. By chance, these new secondary substances can alter the suitability of plants as food for insects. That is, insects may suffer some fitness disadvantage, and the corollary is that plants may be able to reproduce better because they're not getting eaten by these insects. And therefore, plants being released by the constraints imposed by herbivory can undergo population growth, inhabiting of additional niches or niches, depending on what you prefer, and enter what is sometimes referred to as a new adaptive zone. Now, this is kind of a little bit where the magic happens. Ehrlich and Raven didn't really specify very much how this was supposed to happen, and there's been a lot of debate in the scientific literature as to what evolutionary radiations, what the mechanism for causing these evolutionary radiations actually is. But let's leave that aside for now. Now, likewise, by mutation or recombinations, insects can also evolve mechanisms that allow them to overcome these secondary plant substances that were previously dragging them down, causing them to be sick, having lower fitness, and so on. Because any of those insects that just by chance had this now were at advantage over the others in the population that didn't have these traits, and their genes are going to spread in the population. And therefore, they find all these food plants that are available to them that have no competitors on them. And they're able to grow their populations and also speciate on them. They enter these new adaptive zones. And so it's this reciprocal evolutionary adaptive changes that plants and insects can have on each other that allows them to escape herbivory and then diversify, colonize new plants and then diversify in the absence of competitors, and so on and so on. And this is the so-called escape and radiate model of coevolution that Ehrlich and Raven proposed. There's other models, but this is the one we're going to start with today. And again, they implied a couple of different elements in this particular hypothesis, that there was reciprocal selection. Herbivore puts some selective pressure on the plant, and the plant applies selective pressure on the herbivore. That actually is kind of the core strict definition of coevolution. That there's reciprocal adaptation, that is, they both of the partners respond to each other's selection pressure, and that there's a stimulation of these speciation events that allow them to become more species in the absence of herbivory. And that's, as I said before, is a little bit where the arm waving actually happens in all this. The mechanisms of adaptive radiation. This again is the escape. This is the model that Ehrlich and Raven proposed. The escalation and diversification hypothesis is a modification of the Ehrlich and Raven. And I think Thomson was the one who actually coined the escape and radiate terminology that embodied the Ehrlich and Raven approach. So what evidence is there that is consistent with this kind of an hypothesis of reciprocal selective impacts and interactions that could lead to this diversification of plants and insects? Going back to this question of why are groups like beetles so diverse, so speciose? An interesting observation is that over half of the beetles are phytophagous. That is, they feed on plants. And a large number of these herbivorous beetles actually feed on flowering plants. And flowering plants are the group of plants that are the most diverse themselves. So when a classic study by Brian Farrell at the University of Maryland at the time actually looked at the timing of the diversification of plants as inferred through the geologic record, looking at fossils. How many new different kinds of plant species existed as you go back in time? And what was interesting is that right around the Jurassic here, approximately 60 million years ago, there was a very rapid period of expansion, increasing the number of species of flowering plants. And you can see it here. There was also some additional radiation that you can see, or additional speciation rates. There was also a period of increased diversification here during the Tertiary, a much more recent period. This was coincident with the timing of when beetles also started to diversify. So you can start seeing some synchrony between the diversification rates of plants and the diversification rates of insects, of herbivorous beetles in particular. Now, what was interesting, the study that Farrell did, is he actually looked at the fact. What was interesting with Farrell's study is that he noticed that although beetles, for as a group, are a monophyletic group, that is, they all share the same common ancestor, they are not necessarily all specialized on feeding on angiosperms, on flowering plants. Certain groups of the Phytophagous beetles feed on kind of ancient evergreens, the cycads. Some of them feed on conifers. Some of them feed on angiosperms that are dicots. These are the flowering plants that are the most diverse. And some of them feed on monocots, things like lilies and onions and things like that. But one of the key observations that Farrell made is that there are many more beetle groups, beetle genera, that occur in clades, in groups of plants that actually feed on angiosperms, and not as many species or genera, in this case, an indicator of diversification, for those that feed on conifers and cycads that did not have that same great rate of diversification during the late Cretaceous and Jurassic. When Farrell looked at the number of, when Farrell then compared sister groups within the beetles that either fed on non-angiosperms compared to those that fed on angiosperms, the pattern is even more striking. It's a little hard to read like this, but in parentheses here are the number of it's a little hard to read here, but you can see the number of genera that are found within the groups that are here in green, that are non-angiosperms, versus the ones here in purple that are angiosperm feeding. And you can see, without even me showing you, that the groups that had that were feeding on the angiosperms were significantly more speciose than the ones that were feeding on the non-angiosperms. And here you can see another one in this red box here, species in the groups that were not feeding on angiosperms, 8 and 18, compared to ones that have 3,000, 300, 2,000, 8,000 species in those groups. So when you look at the groups, this is a simplified version. This is a simplified version right here. Here's the groups that feed on angiosperms, much more speciose than the sister groups that were feeding on the non-angiosperms. And this was repeated multiple times over the evolutionary phylogeny of beetles. Again, lending some support to the idea that association with flowering plants that were undergoing rapid speciation was also associated with rapid speciation of the hosts themselves. So certainly consistent with the idea of adaptation and radiation that we see that Ehrlich and Raven had hypothesized. So what about the other components of the Ehrlich and Raven system? That is, that there was an escalation of defensive chemistries. Now, mind you, the terminology here was very typical of the time. Ehrlich and Raven were reading and working and writing about these topics during the Cold War era. So this is idea of escalation of defenses that governments were doing in response to each other. If the Russians have 3 nukes, well, then we'd better have 5. And if the Americans have 5, then we better have 10, and so on and so on. The same kind of mentality was brought to bear on these ecological systems here. Well, one person that made some incredible insights and inroads in this particular area was Professor May Berenbaum. May was a just starting faculty member at the University of Illinois. I'm showing her here in a picture from 2014 next to President Barack Obama when she received the Presidential Medal of Science, in large part because of her contributions to our understanding about the evolution and diversification of insects. But at the time, she was a young professor at the University of Illinois. I had the great fortune to actually take her insect ecology class when I was an undergraduate with her in the late 80s. And May made the observation that there was a relationship between the toxicity and the complexity of certain secondary compounds in that are present in some plants, and the association of caterpillars, butterflies that fed on those particular plants that had those compounds. And these compounds belong to a group that shared the same general motif here that started with this very simple carbon aromatic ring with a couple of oxygens right here, a molecule called coumarin. A hydroxylation of this position here and adding of this group here makes them hydroxycoumarins. You can then loop this piece here with a couple of more carbons to make a linear furanocoumarin. Now there's a furan ring, which is this thing here, and it's in this linear position. And other plants, on the other hand, flip this ring over so that it kind of closed on this side, so that it's a so-called angular furanocoumarin. So these were compounds that were starting to be mapped and discovered and then mapped in plants. And May made the observation that the hydroxycoumarins, which were maybe the simplest way that you can kind of take that original motif of coumarin and make it a little more complex, was actually present in a lot of different families, 3 dozen families, including plants like licorice here in the bean family, lavenders, which are in the mint family, the rubiaceae here, the bedstraw family, and lots of different butterflies and their caterpillars actually feed on these chemicals. You might even recognize this one here, warfarin. Warfarin is a blood thinner that is used for treating people with strokes or heart attacks, and it keeps the blood flowing so it doesn't clot in your arteries. And if you recognize here, WARF is actually one of the buildings that we have here on campus, the Wisconsin Alumni Research Foundation building. This was a compound that was actually patented and isolated and patented here at the University of Wisconsin. If we look at the distribution of linear furanocoumarins, so this is the hydroxy group that now has been flipped over right here, you only find it present in about a dozen plant species. Primarily, one of the most common plants that has these is poison hemlock. So linear furanocoumarins are toxic to humans as well. Poison hemlock was what Socrates drank a brew of to commit suicide back in the Greek times. Don't drink poison hemlock, don't eat it. On the other hand, species like the black swallowtail, this is an old world swallowtail present in Europe, part of the Macaon group, has the ability to feed on these plants. And again, mind you, it's the caterpillars that are doing the feeding. The adults don't do all that much but nectar and lay their eggs. And then one of the interesting things about linear furanocoumarins here is that in the presence of light, they have the ability of cross-linking the base pairs that are present in DNA. So here's one of the base pairs here. Remember, they have the T, C, G, A base pairs that form the codes of our genetic signals. And with the presence of these, and what pheranocoumarins can do is actually bind to them and actually lock themselves into the double-stranded piece of DNA, preventing replication and translation from actually happening. And so these can be actually quite damaging to DNA, this cross-linking phenomenon, and quite toxic as a result. And angular pheranocoumarins also have this property. And the angular pheranocoumarins are only present in 3 plant families: the Apiaceae, the Rutaceae, and the Fabaceae. The Apiaceae, again, are things like carrots, parsnips, like this, hemlocks. Used to be the umbellifer family. They have these umbel-like flowers here. The Rutaceae is citrus. The Fabaceae are the legumes, the bean family. And as you can imagine, there are some herbivores like these short-tailed swallowtails here that can feed on these particular plants. Now, what's interesting here is that what's interesting in these patterns that Dr. Berenbaum was able to document is that you have a class of compounds that are relatively common, that occur across a lot of species, and then there were derivations of that chemistry that were simply dependent on some enzymes that could kind of flip the bonds of those particular hydroxy groups that created more and more toxic derivatives, and fewer herbivores were able to colonize those plants and persist on them. Again, consistent with the ideas of the Ehrlich and Raven. So the Ehrlich and Raven, consistent with the ideas of the Ehrlich and Raven hypothesis. Now, one of the neat things that May and her group was able to do is that they actually found that the groups of caterpillars that actually were able, the groups of butterflies who were able to detoxify some of these compounds, had a class of detoxification enzymes in a family called the cytochrome P450s. These are membrane-bound gut enzymes that have the capacity to actually break off or to put on actually water, that have the capacity to put on hydroxyl groups onto these molecules to make them more water soluble and get them out of their body quickly. And this xanthotoxin metabolism, xanthotoxin is one of those pheranocoumarins there, varies across caterpillar genotypes. So some genotypes are really able to detoxify them, whereas others are not able to detoxify them very much, suggesting that they have a genetic basis. So here now, there's a mechanism, a detoxification enzyme, that is able to overcome those defenses and has the potential to be under selective advantage. These families here should be at a greater advantage compared to these families here. And if that association is very strong, those traits will become more widespread in the population. And so kind of in a classical interpretation of Berenbaum's scenario, there was again the ancestral form of the coumaric acids, the hydroxyl groups attached to the coumarins, the creating of the furan group onto the coumarin, and then the angular form of that, which made them more and more toxic. And whereas most papillionids could deal with the coumaric acids, fewer and fewer species could actually deal with the more derived forms like the angular pheranocoumarins. Now, there's been a lot of work done. Since the late 80s on this particular topic, even with the particular system that Dr. Berenbaum worked on, studying the mechanisms, and proposing alternative hypotheses that actually may create the same kind of pattern as you may see here. And we're going to get to that in a little bit, and kind of a critique of some of the data that supports a critique of the data that is used to support this model of coevolution between herbivores and their plants. But let me give you one more example, which is a very nice example here. This was a study done by Judith Becerra. She was let me give you another example here of a very nice study. Let me give you another example here of a very nice study done by Judith Becerra at the University of Arizona. And she was studying a group of flea beetles. These are chrysomelids in the genus Blephariida. And these are exist throughout the Old World tropics, kind of Central Africa, but also in the New World tropics. And they feed on a group of leguminous plants in this genus Bursera. And one of the neat things about these Bursera species is they're chock full of terpenoid compounds, and you heard about that in our previous lesson. And some of the plants actually keep these compounds under very high pressure, such that when the veins are cut, they will actually squirt out, kind of like a fire hose, the compounds at whatever it is that was feeding on it, kind of shooting a mouthful of kind of caustic, nasty chemicals into their face. And here you can see one of these kind of squirting Bursera species here. And these caterpillars have evolved a series of tricks to kind of overcome these particular defenses. Some are behavioral, and some are actually physiological as well. And what Becerra was able to do was actually develop a phylogeny, an evolutionary, a reconstructed evolutionary history of the beetles here, the Blepharid herbivores. And again, each one of these branching nodes kind of shows a family of related species. And these are the most recent common ancestors. And she was also able to kind of reconstruct the time of the split based on a molecular clock. And she did the same for the plant groups, and she did the same for the plant phylogeny, where the Bursera evolutionary history, was kind of derived from some information based on their DNA. And what you can see here and one of the things you can see here in the red and in the pink are groups of Bursera that have the squirting traits. And the ones in the green here are the ones that have these complex chemistries of these nasty terpenoid compounds. And they're in two very different groups within the genus Bursera. There's one that's over here, and there's one that's over here. And this species here that knows how to deal with these squirters is related to the ones that know how to that have evolved ways of actually that have evolved ways to actually detoxify the nasty compounds that are present in some of these more derived lineages here. And here are some actually that have these behavioral traits that allow them to trench the leaves so as to overcome the squirting pathways that some of them have. And what you can see here is that there's a lot of convergence and crisscrossing of these lines, suggesting that there is not necessarily a strict evolutionary kind of lockstep changes between the plants and the insects, but there are traits that then allow certain groups that have kind of unlocked unlocked the mechanisms for how to overcome those defenses that allowed them to colonize all of these different species here. So back to the Ehrlich and Raven scenario, if there is this continual evolution or coevolution between insects and plants, what you would expect is that there could be a variety of ways in which plants could escalate their defenses. They can either increase the concentration of the compounds. They can either have more different kinds of compounds that would make them more toxic as an ensemble. They might actually have compounds coming from very different biosynthetic pathways. So imagine ones that have very different modes of actions that would challenge the detoxification machinery of the insects much more so. And they could produce completely novel compounds or strategies that are new to the group as a whole, or all of those things. And the longer this interaction takes place between these herbivores and these plants, the more of these you would expect to get over time. And what Becerra showed was actually if you use the number of branching points as an indicator of how old an association may be, how many times has kind of diversification actually happened, whether you're looking at the bean family or at this aster family, that there seems to be an association between the time of relationship between the plants and the insects, and things like the number of different types of alkaloids. These are nitrogen containing compounds, or the diversity of those compounds over time, like this. And the same thing here for plants and their defenses in the Fabaceae, and the same thing here for plants for the number of compounds and the diversity of those compounds for plants in the Fabaceae. So the longer they're in association with those specialized herbivores, the more likely you get this diversification of these chemistries. So taken together, that the mechanisms that were proposed by Ehrlich and Raven are actually observed in some of these lineages, that there is more rapid diversification of lineages of both insects and plants, and that there's fewer and fewer insects that have been able to kind of overcome these defenses, these more specialized defenses, all point in this direction that yes, there's something special about the interaction between insects and plants that allow them to reciprocally respond to each other, and this may have an effect on the rates of speciation between them. Now, one of the big critiques of these kinds of studies is that just because you see a pattern that makes it look like the phylogenies, the evolutionary histories of the insects kind of match very neatly to the evolutionary history of the plants, does not necessarily imply causation. And causation is really the key thing that's involved in our understanding of whether coevolution is happening, whether there's reciprocal evolutionary responses of an insect to a plant. And in fact, speciation may just be incidental to the fact that these organisms may be occurring in the same area. Maybe they colonized a particular place after some kind of vicarious event. Maybe there was a ridge of mountains or a lake or some kind of division, a large body of water that prevented one population from interbreeding with another one. And that in and of itself is what created divergence of these lineages so that they became reproductively isolated and could not interbreed and therefore became new species. And that plants and insects did this independently, and it just so looks like they have these same patterns because they did it in response to a third variable that had nothing really to do with the interaction between them. So this is the so-called kind of allopatric model of speciation, where divergence happens because there's just lack of reproductive opportunities between these two populations that, given enough time, thousands, millions of years, become reproductively self-incompatible, and that's when you get the evolution of new species. And so for coevolution to happen, there needs to be causality. This says causal, not casual here. So it's causal co-speciation is really what you need for coevolution, not incidental co-speciation, which is just the speciation because two things happen to be in the same place responding to the same environment, but not really because of the interactions between them. And really, one of the challenges of kind of studying this macro level patterns is that causality is very difficult to actually suss out. So in the next lecture, what we're going to look at is what is some of the evidence that points to the possibility that some of these genetic changes could actually be happening in ecological time.